Deep Dive Listening. This video provides an easy-to-understand English explanation of Dr. Sakaguchi et al.'s research on regulatory T cells and FOXP3, for which they were awarded the Nobel Prize in Physiology or Medicine on October 6, 2025. This video is intended for English listening practice only and is not meant to provide medical advice. While we strive for accuracy, we do not guarantee the completeness or correctness of the information presented. Now, let's get started. On October 6, 2025, the Nobel Assembly at Karolinska Institute in Sweden announced that the year's Nobel Prize in Physiology or Medicine would be awarded to three scientists: distinguished Professor Shimon Sakaguchi of Osaka University and U.S. researchers Dr. Mary E. Brunko and Dr. Fred Ramsdell. The prize was awarded for discoveries concerning peripheral immune tolerance. These discoveries elucidated how the immune system avoids attacking the body's own tissues. Dr. Sakaguchi and colleagues discovered regulatory T cells, Tregs, which suppress excessive immune responses in the body. This provides a key answer to how we can sustain life without attacking self. This discovery not only advances basic science, but also has potential to lead to new therapies for many conditions, including autoimmune diseases such as rheumatoid arthritis and type 1 diabetes, allergies, cancer, and transplant rejection. Osaka University President Atsushi Kumanogo stated, "Modern medicine cannot be discussed without regulatory T cells," underscoring that this discovery forms one of the foundations of contemporary medicine. This video is organized into 20 chapters to make the discovery accessible to non-specialist viewers. It explains the basics of the immune system, the path to the discovery, and how it may influence future medicine. The human body is constantly exposed to threats such as viruses, bacteria, and abnormal cells that arise within the body. The biological system that defends the body against these threats is the immune system. The term immunity means the state of being protected from disease. The immune system has two main roles. First, it detects and eliminates pathogens such as viruses and bacteria that invade from outside the body. Second, it disposes of cellular debris and abnormal cells that arise inside the body, including those caused by errors in DNA replication, such as cancer cells. In this way, the immune system maintains health by distinguishing external invaders from internal abnormalities and removing them. The immune system is not a single mechanism. It is broadly divided into two arms: innate immunity and adaptive immunity. Each has distinct roles, and they work together to defend the body. Innate immunity is a defense system that is present from birth. Cells such as macrophages, neutrophils, and natural killer NK cells constantly patrol the body and, upon detecting foreign material, such as pathogens, they attack immediately regardless of the specific type. It is the body's first line of defense that responds at the earliest phase of invasion. However, some formidable pathogens cannot be handled by innate immunity alone. In such cases, the second arm, adaptive immunity, comes into play. This is a more specialized defense that can recognize and remember features of particular pathogens, called antigens. The hallmark of adaptive immunity is immunological memory. For example, after a measles infection, people are much less likely to get sick again. That is because adaptive immunity remembers measles virus antigens and mounts a much faster and more potent response upon re-exposure than during the first encounter. Vaccines harness this mechanism of immunological memory. By administering in advance an inactivated or attenuated pathogen, or components of it, vaccination generates memory against a specific pathogen without causing the disease. In this way, close coordination between innate and adaptive immunity enables the body to cope with diverse threats. One of the immune system's most important abilities is to accurately distinguish enemies that should be attacked from the body's own self, which must be protected, among the approximately 37 trillion cells that make up the body. Without this discriminating ability, the immune system would attack its own normal cells and organs. To make this distinction, cells display substances called antigens on their surfaces. As they circulate through the body, immune cells continually inspect these antigens and decide whether they originate from self cells or from enemies, such as viruses and bacteria. They target only those identified as enemies. This distinction between self and enemy is a fundamental principle of the immune system. However, for various reasons, this discriminating function can malfunction, and the immune system may attack the body's own tissues. This condition is known as an autoimmune disease. Explaining why this discrimination sometimes breaks down lies at the core of the research recognized by this year's Nobel Prize. In adaptive immunity, a central role is played by a type of lymphocyte called a T cell. After being produced from hematopoietic stem cells in the bone marrow, T cells mature in an organ in the upper chest near the heart called the thymus and acquire specialized functions. The T in T cell is derived from the thymus. T cells can be classified into several types according to their functions. One representative type is the helper T cell. These cells recognize foreign substances, antigens, that have entered the body and transmit that information to other immune cells, issuing instructions that activate immune responses. Without signaling from these cells, efficient immune responses do not occur. Another type is the killer T cell. Receiving instructions from helper T cells and others, these cells directly recognize virus-infected cells and cancer cells and destroy them. Thus, T cells are powerful cells that serve as the core of the immune system. However, because their functions are potent, if they are not properly controlled, excessive responses can harm the body. Accordingly, how such powerful T cell activity is controlled was an important unresolved question in immunology at the time. Autoimmune diseases are a group of disorders in which the immune system, whose role is to defend the body, mistakenly attacks the body's own normal cells and tissues. They arise because, for various reasons, the immune system can no longer properly distinguish self from non-self and misidentifies components of the self as targets. As a result, the targeted tissues develop chronic inflammation and damage occurs in various organs. For example, when the immune system attacks the joints, it causes rheumatoid arthritis, leading to joint pain and deformity. If the insulin-secreting cells in the pancreas are destroyed, type 1 diabetes develops, and lifelong insulin therapy is required. There are also severe diseases such as systemic lupus erythematosus, in which the targets are not limited to a single organ, but can involve the entire body, including the skin, kidneys, heart, and brain. Many of these disorders are difficult to cure with current medicine and tend to follow a long-term course with repeated periods of remission and flare-ups. Therefore, in addition to physical symptoms, patients bear a psychological burden from not knowing when their disease might worsen. Accordingly, elucidating why the immune system targets the body's own tissues has long been a major challenge in medicine. In efforts to understand the mechanisms of autoimmune disease, immunology at the time relied on a basic premise. Namely, T cells capable of attacking self-tissues are screened during maturation in the thymus, and most of them are eliminated. This selection process in the thymus is called central immune tolerance. However, as research progressed, it became clear that a small number of self-reactive T cells that slip through thymic selection are present in the body. This fact raised a new question in immunology. Why do these potentially self-reactive T cells, after passing selection in the thymus and circulating throughout the body, the periphery in technical terms, usually not attack self-tissues? If selection in the thymus were the only control mechanism, it would not be surprising for T cells that passed it to attack self-tissues somewhere in the body. However, in healthy individuals, such events are rare. This observation suggested that there is another mechanism outside the thymus, namely in the periphery, that suppresses excessive self-reactive immune responses. This previously unresolved second control mechanism is peripheral immune tolerance. Demonstrating the existence of this mechanism and identifying the elements that play a central role were the goals of Dr. Sakaguchi and colleagues' research and form the core achievements cited for the Nobel Prize. At a time when many researchers were focused on boosting immune responses, that is, making them more aggressive, Dr. Shimon Sakaguchi took a different view. Having graduated from the Faculty of Medicine at Kyoto University in 1976, he was drawn to the fundamental immunological question of what is self. He reasoned that a system as powerfully aggressive as immunity must also include mechanisms that appropriately dampen, that is, suppress its responses. His hypothesis was that offensive functions alone are insufficient to maintain physiological homeostasis. Specifically, he proposed that among T cells there exist not only cells that promote immune responses, but also cells that suppress them, and that health is maintained by the balance between the two. His focus on suppression, a concept that was not mainstream at the time, became the starting point for his later discoveries. Dr. Sakaguchi began investigating unknown factors that restrain excessive immune reactions. Before the 1980s, when Dr. Shimon Sakaguchi began to pursue his research in earnest, a controversy existed in the field of immunology. In the 1970s, the concept of suppressor T cells that suppress immune responses, was proposed, and, for a time, it became a major focus among researchers worldwide. However, in the 1980s, researchers were unable to conclusively identify the entity of suppressor T cells, and reproducible experimental data remained elusive. Furthermore, advances in molecular biology indicated that the proposed mechanisms for suppressor T cells were theoretically untenable, and interest in the concept rapidly waned. As a result, the prevailing view in the academic community at the time became that suppressor T cells do not exist. Dr. Sakaguchi published his first paper suggesting the existence of cells that suppress immune responses in 1985, precisely overlapping with the period when the suppressor T cell concept was being rejected. Consequently, many researchers regarded his work as pursuing a previously discredited idea, and it was not fairly evaluated. Nevertheless, Dr. Sakaguchi believed that even if prior theories were flawed, the phenomenon that cells exist in the body that regulate immunity remained true. Guided by that conviction, he continued his research despite receiving little recognition from the academic community. The stagnation in this line of research reached a turning point in 1995. Dr. Shimon Sakaguchi published a landmark paper in immunology. Using mouse models, he experimentally demonstrated the existence of a specific T-cell subset that suppresses immune responses. The key to this discovery was a cell surface molecule called CD25, the IL-2 receptor alpha chain. Sakaguchi isolated the T-cell subset that expressed high levels of CD25 and analyzed its function. He found that this CD25 positive population potently suppressed the activity of other, more aggressive T cells. Furthermore, when CD25 plus cells were removed from CD4 plus T cell suspensions, and the remaining cells were transferred into athymic nude mice, the recipients developed severe autoimmune disease. This provided decisive evidence that this cell population restrains the spontaneous development of autoimmunity under normal conditions. These cells were later named regulatory T cells and are commonly abbreviated as Tregs. Tregs prevent other immune cells, such as killer T cells, from overreacting and attacking the body's own tissues. Subsequent studies showed that in healthy individuals, roughly 5 to 10% of peripheral CD4 plus T cells are Tregs. This discovery clarified the identity of the long-suspected suppressive immune cell population by defining it as Tregs. Around the same period when Dr. Sakaguchi demonstrated the existence of Tregs, a U.S. team led by Dr. Mary E. Brunko and Dr. Frederick Ramsdell pursued related research using a genetic approach. They studied the scurfy mouse, which is born with a severe, fatal autoimmune disease. Through genetic analysis, they identified that the disease in these mice is caused by a defect in a single gene. They showed that loss of function of this gene, which they named FOXP3, triggers abnormal activation of the immune system. Furthermore, in humans, a rare inherited disorder with symptoms similar to the scurfy phenotype, known as IPEX syndrome, was found to be caused by mutations in the same FOXP3 gene. In 2003, Dr. Sakaguchi's work connected these previously independent lines of evidence. He demonstrated that the Tregs he had identified specifically and abundantly expressed the FOXP3 gene. This established FOXP as a master gene essential for the differentiation and function of Treg cells. Thus, by integrating findings from different research approaches, it was demonstrated that FOXP mutations cause Treg dysfunction, leading to severe autoimmune disease such as IPEX. As a consequence, at the genetic level, it became established that Treg cells are indispensable for sustaining life. By integrating these discoveries, the overall picture of peripheral immune tolerance, a long-standing problem in immunology, comes into view. The key question was, why do potentially self-reactive T cells that have passed selection in the thymus, central, not attack self-tissues throughout the body, periphery? The answer lies in peripheral immune tolerance, the second control mechanism in which the regulatory T cells, Tregs, discovered by Dr. Sakaguchi and colleagues, play a central role. After differentiating in the thymus, Tregs circulate throughout the body via the blood and lymph. When they detect T cells attempting to attack self-tissues, they prevent damage by releasing suppressive cytokines and by direct contact-dependent mechanisms that restrain those cells. In short, the human body is protected from autoimmunity by a two-tiered control system. The first tier is the elimination of self-reactive T cells in the thymus, namely central immune tolerance. The second tier, which controls cells that escape this selection, is peripheral immune tolerance, mediated by Tregs. Because these two tiers exist, the immune system can mount strong responses against external threats while sparing self-tissues and maintaining proper balance. Clarifying the existence of peripheral immune tolerance, indispensable for sustaining life, and the roles of Tregs and FOXP3 at its core, constitutes the principal achievement of Dr. Sakaguchi and colleagues and is central to the Nobel Committee's reasoning for the award. Although Dr. Sakaguchi published the paper that proved the existence of regulatory T cells, Tregs, in 1995, he received the Nobel Prize in 2025, leaving a 30-year interval between the discovery and the award. Understanding why this temporal gap arose is important for appreciating the pathway by which scientific discoveries come to be publicly recognized. The first reason is the long-term impact of the controversy over suppressor T cells, discussed in Chapter 9. Because Dr. Sakaguchi's finding resembled a concept that the scientific community had once concluded did not exist, it took time for its significance to gain broad acceptance. To establish the validity of the discovery and secure scholarly recognition, replication by many researchers and the accumulation of corroborating evidence were required. The second reason relates to the Nobel Prizes selection criteria. The Nobel Prize tends to be awarded only once it is clear that fundamental research has conferred a benefit to humankind through practical impact. It was necessary to demonstrate through clinical trials that the discovery of Tregs could lead to effective therapies for autoimmune diseases and cancer. In recent years, therapies targeting Tregs have advanced into clinical testing, and the emergence of promising results is thought to have been an important factor in the award. The third reason is that it took time for the magnitude of the discovery's impact on science as a whole to become clear. The discovery of Tregs substantially reshaped the conventional understanding of immunology. Building on this new concept, numerous lines of research were initiated and advanced in related areas such as tumor immunology, transplant immunology, and allergology. The establishment of this wide-ranging scientific influence led to this year's award. The significance of the award to Dr. Sakaguchi and colleagues goes beyond the discovery of a new cell type. It changed the basic conceptual framework of immunology and provided a new perspective for understanding life. The first shift was that the field's primary focus expanded from attack to also include control. Before the discovery of Tregs, immunology mainly focused on how to efficiently attack and eliminate enemies. However, the discovery of Tregs revealed a new foundational principle. The immune system's normal function is maintained not by attack capacity alone, but by a balance between attack and suppression. The second shift was that understanding of where immune tolerance is established expanded from the central to the peripheral. Previously, self-tolerance was thought to be established mainly in the thymus, central, during T-cell maturation. However, the discovery of Treg-mediated peripheral immune tolerance showed that, even after leaving the thymus, the balance of immunity is continually adjusted at various sites throughout the body in a dynamic process. Through these two major conceptual changes, our understanding of the immune system shifted from a predetermined, static model to a more complex, dynamic system that is constantly fluctuating and being adjusted. The achievement of Dr. Sakaguchi and his colleagues was to lay the foundation for this new understanding. The most important impact of the discovery of regulatory T cells, Treg, on medicine is that by artificially controlling their function, it has enabled the development of new therapies for diseases that were previously difficult to treat. These therapeutic strategies fall broadly into two distinct approaches. The first approach is to increase the number of Treg, or to strengthen their function. This aims to suppress states in which the immune system overreacts. The main targets include autoimmune diseases that attack self-tissues, allergic diseases such as pollen or food allergy, and rejection after organ transplantation. The second approach, the reverse of the first, is to reduce the number of Treg or weaken their function. This seeks to release brakes on the immune system and enhance its inherent effector capacity. The principal target is cancer, which can evade immune surveillance. Accordingly, the concept of adjusting Treg function, either augmenting or inhibiting it, has the potential to become a key therapeutic strategy in clinical practice. Research is pursuing disease-tailored control, increasing Treg to restrain excessive immune responses, and decreasing Treg to boost anti-tumor immunity. Therapeutic approaches that aim to increase the number of regulatory T cells, Tregs, are being investigated as a potential new option, particularly for the treatment of autoimmune diseases. Rheumatoid arthritis is a representative autoimmune disease with an estimated 600,000 to 1 million patients in Japan. Conventional mainline treatments include biologic agents that suppress the overall function of the immune system. While these drugs are highly effective, they lower overall immune function and thereby raise the risk of infections, such as pneumonia, and their high cost is also a challenge. Research has shown that dysfunction of Tregs contributes to RA pathophysiology, and increasing Treg numbers or restoring their function could act directly on the disease cause and potentially offer therapies with fewer side effects. However, it has also been pointed out that under inflammatory conditions, Tregs may convert into cells that promote inflammation, and research is underway to establish safe and effective therapies. Type 1 diabetes is a disease in which the immune system destroys the pancreatic beta cells that produce insulin. In Japan, the number of patients is estimated at about 100,000 to 140,000, and many patients need to self-inject insulin several times a day for life. Therapies using Tregs aim to suppress the immune attack on these beta cells itself. This approach may protect residual beta cell function in patients at disease onset and help slow disease progression. In addition, combining it with regenerative medicine that transplants beta cells produced from iPS cells and the like could enhance treatment by preventing the transplanted cells from being destroyed by the immune system. Dr. Sakaguchi has indicated an intention to begin clinical trials in this field within 3 years. Strategies to increase regulatory T cells, Tregs, are also being explored for application in organ transplantation. Patients who receive transplants of organs such as the heart, liver, or kidney face the problem of rejection after surgery. Rejection is the phenomenon in which the immune system recognizes the transplanted organ as foreign and attacks it. To prevent this, patients must continuously take immunosuppressive drugs that dampen overall immune function. However, these drugs carry serious side effects, including increased risks of infections and cancer. If we could artificially induce immune tolerance, a state in which immune responses are suppressed only against the transplanted organ, patients might be able to discontinue immunosuppressants. Tregs are thought to play a central role in achieving this goal. This approach has already advanced to clinical studies. In Japan, an investigator-initiated, multi-center Phase 1/2 trial led by Juntendo University is evaluating JB101 in living donor liver transplantation. Participating sites include Hiroshima University Hospital. The trial's primary efficacy endpoint is operational tolerance, defined by sustained cessation of immunosuppressants. In a prior Japanese pilot study of Treg-based cell therapy in living donor liver transplantation, 7 of 10 patients were successfully weaned off all immunosuppressive medication while maintaining graft function. These results suggest that Treg-based cell therapy could transform how rejection is controlled in transplantation. Strategies that reduce the number of regulatory T cells, Tregs, offer a new approach to cancer therapy. Cancer cells employ various mechanisms to evade attack by the immune system. One of the key mechanisms is the exploitation of Tregs. Studies have shown that immunosuppressive Tregs accumulate in large numbers in and around many tumors. By actively recruiting Tregs, tumor cells establish an immunosuppressive microenvironment around the lesion. As a result, the function of cytotoxic killer T cells that should directly attack cancer cells is suppressed. Indeed, multiple reports show that higher intratumoral Treg accumulation correlates with poorer patient prognosis. Based on these insights, new cancer immunotherapies that target Tregs are being developed. The basic concept is to remove Tregs in the tumor milieu or inhibit their suppressive functions. This is expected to reactivate previously suppressed killer T cells so they can attack cancer cells. This approach may yield greater therapeutic benefit when combined with widely used immune checkpoint inhibitors, which release the brakes on killer T cells. Clinical trials worldwide are testing Treg-targeting agents in combination with checkpoint inhibitors, aiming to maximize anti-tumor immunity and establish new options for cancers that have been hard to treat. Technologies that modulate regulatory T cell, Treg function, are expected to find applications not only in severe diseases, but also in more familiar health problems. Allergic diseases are one example. Conditions such as hay fever, food allergy, and atopic dermatitis are triggered when the immune system overreacts to substances that are normally harmless, allergens. This too is a form of immune imbalance. By enhancing Treg function, therapies are being developed to suppress this excessive response and to improve the allergic predisposition itself. In the future, preventative use is envisioned, for example, taking a Treg-expanding drug before a particular season to avert symptom onset. Tregs are also deeply involved in maintaining pregnancy. Because the fetus carries paternal genes, the maternal immune system can, in part, recognize it as non-self. Yet, in most cases, an immune attack does not occur. One reason is the accumulation of Tregs around the placenta, in the decidua, which suppresses aggressive immune responses against the fetus. Detailed elucidation of this mechanism, called maternal fetal immune tolerance, may help uncover causes of recurrent miscarriage and lead to new infertility treatments. Thus, research on Tregs offers a new therapeutic approach, rebalancing the immune system for many conditions that have traditionally been considered constitutional or of unknown cause. This suggests the potential to fundamentally change both how diseases are treated and how health is maintained. The 2025 Nobel Prize in Physiology or Medicine did not recognize only the discovery of a particular cell called the regulatory T cell. Rather, it honored a body of research that elucidated the dynamic balance between immune attack and suppression that maintains self-tolerance, and pointed new directions for the future of medicine. This discovery emerged from the work of a scientist who continued to pursue his own hypothesis that cells exist which suppress immunity, despite prevailing views in the field to the contrary. As Dr. Sakaguchi himself has noted, the pursuit of scientific truth is not an easy process. Even during periods when the work was hard to evaluate academically, sustained research has led to today's expectations for new therapies against many diseases. The trajectory of this research not only illustrates the benefits that scientific progress can bring, but also underscores the importance of a researcher's long-term commitment to a single problem and the conviction to pursue truth. The discoveries by Dr. Sakaguchi and his colleagues are expected to influence future researchers and continue contributing to humanity's efforts to overcome disease.